Hello, happy Wednesday, March 3rd, 2021. We are concluding Module 2 this week. We've done a little bit with muscle physiology. Last week was DNA and RNA. This week we'll look a little bit at immunity. March 7th, which is Sunday, is when you want to try and have everything from Module 2 completed. 3 Padlets and the Padlet surveys, of course, 3 case studies, 3 PhysioExes, and 3 exams. So that puts us at the halfway point come Sunday night. So excellent work. I'm in the process of grading. I'll continue to grade today into tomorrow and certainly throughout the weekend, as I know many of you will continue to be handing things in. Let's see. Couple of things. Module 3 and Module 4 are both open. Now, Module 3, I may have mentioned this last week, but you're going to see, and next week we'll start Module 3, so I'll dive in deeper. But in case you're working ahead, this is not a typo. There are 4 PhysioExes in Module 3, but only 2 in Module 4. So you won't have a Unit 12, which is the last unit, PhysioEx, but you do have 2 you have an extra PhysioEx in Module 3, Unit 7. So anyway, we'll talk about that more next week. But no, those are not typos. But if you are working ahead, know that there are a couple of extras in Module 3 and one less in Module 4. So it all evens out in the end. The other thing is I will no longer be meeting live on Wednesday nights. I will be doing these pre-recorded video sessions for you. All of the so I'll be going through the Padlets, case studies, PhysioExes in these videos each week. And then I'll also have the video link with which you also have in your Blackboard. Again, if you go under module over here, the module Power Video Lectures and PowerPoints, you'll find the pre-recorded videos of the lecture material. So these pre-recordings that I'll put out each week will have the, again, the unit Padlets, case studies, discussion, and the PhysioEx demo. So anyway, so that's what's going on there. If you do need to meet with me live, I'm certainly more than happy to do that with you. Just send me an email and we can find out a time. I have quite a bit of availability to do that. So, again, if you do need to meet live, please let me know. And again, my office hours are 12 to 5 on Wednesdays. But again, we can meet anytime if those times don't work for you. I've enjoyed seeing all of you every Wednesday night. So I hope those evenings were helpful. These are going to be just as helpful. They're going to be the same thing. So you can watch them at your leisure this time. Now, those of you watching that are in my Elkhorn Wednesday night group, there are 7 of you. So please check the announcement in your Blackboard. I also sent an email. We will be meeting a couple of times, 4 to be exact, in Elkhorn to kind of finish the semester out. So next Wednesday, March 10th, we'll be in Elkhorn, and then 2 weeks after that. So we'll meet every 2 weeks starting next week. So I'll send out reminders to you Elkhorn students as well. And again, if you have questions, just send me an email. All right, well, let's get to it. We're in Module 2, the very end, getting into immunity. So Unit 6, as usual, it's the same by now, right? We're at midterms. I would think by now you kind of know the routine. So again, you can find your hyperlinks to your Padlets down here. And again, those will take you, one takes you out to the Padlet itself, and the other one is going to be the little survey that you need to complete. So this week's Padlet has 5 videos. Again, you're only required really to watch one, but please watch all 5 as we go throughout the semester. Again encourage you. These videos are extremely helpful. They answer a lot of the basics regarding what we're talking about each week. So, and they're relatively short, but packed with some really good information. And I've checked, and again, if they don't work, please let me know. I have checked the rest of the semester's Padlet videos. All of them are in working order, so that's a good thing. So again, if you do run into any issues, please let me know. But I checked today, and all of them appear to be working. Okay, so that's the Padlets for this week. And then, and again, they do go through the objectives for this particular competency. Okay, and then the case study assignment. Again, with the case study assignment, when we look at that, we do see that there it is, case study 6. And again, I'm just going through the hyperlinks. I think that's the best way to get to where you need to go. And again, under modules and units, you're going to find these as well. You'll find a little more info regarding some of the other Quizlets and some of the links to other websites. But when it comes to the assignments, these hyperlinks off to the side, I think many of you have found to be useful. So anyway, you click that, it'll open up the case study questions. And again, that's important. Obviously, that's what you're going to be turning in. But the questions do, I don't want to say differ, they are amended slightly from what's in the actual case study. So, like there's oftentimes there'll be a table in the case study. So you're going to want to go through and recognize that you don't need to fill out that table. If we were in a face-to-face session, that's something we would do as a group. And then part 5, you don't need to do part 5. And then I added a number 4 in part 4. So we'll go through those here in just a few minutes. And again, how do you find the actual case study? That's going to be down here under COVID-19 treatment for this particular one. The PDF version is going to be the case study, the actual case study. This is the Word doc version. And then of course, this is the Google Doc. So this hyperlink and this first one are going to be the same thing. This one will again open directly and download a Google Doc, which you can type right into. And then the COVID-19 treatment, that's going to be the actual case study. All right, let me do a little bit of adjusting on my screens here. There we go. Pull that up over just a hair. Okay, so obviously at this point, it's just about the 1 year kind of anniversary, if you will, of when certainly when Gateway, the state of Wisconsin as well, was really starting to recognize COVID-19 as a potential issue. So I want to say it was by around March 15th, the Ides of March, right around March 15th or 16th of 2020 is when Gateway pretty much said, we're done. We're going to go online. State of Wisconsin started mandating the masks and the social distancing. So believe it or not, it's been about a year. I think many of you probably don't find that hard to believe. Maybe it was a really fast year for you, maybe a slow year, but I think we can all agree that it was a very unusual year. So required a lot of flexibility and a lot of adjustments to be made in our lives, particularly with school. Many of you had maybe never really done much in the online setting, and now here you are a year later, and you guys are old pros at it at this point. So again, kudos to all of you for being able to adjust and make those types of growth types of changes that needed to be made during a time that, again, unlike any other probably really in the history of certainly modern man. So, so anyway, we're living history even without COVID, right? We're living history, but at this point in the game, I think all of us are pretty well aware of what COVID-19 is, at least on the surface. So this particular case study goes through. We've got a couple of gals who want to hang out with their buddies, and so and they were possibly exposed. One of the moms and co-workers tested positive, and the kids were at the workplace. So anyway, it just kind of paints this picture and of kind of what we you know, uh, knew, didn't know. I mean, just, regarding COVID, this is pretty early on in the game. So not a heck of a lot was known right away. But I think as you go through this case study, you'll see how well it certainly applies to today. And certainly as you reflect on the past year. So anyway, just ask what are the symptoms of COVID-19? It's a laundry list potentially, right? So some of the typical viral infection symptoms that we see regarding fever, coughing, shortness of breath. We've heard a lot of folks talking about a loss of smell, loss of taste. Some of you maybe have been exposed to COVID yourselves and experienced it, or at least have had maybe close friends, family, or colleagues, patients that have had COVID. So you can add anything else in there. These don't specifically have to be CDC or it could be what you've experienced as well. Yeah, World Health Organization certainly too is a good reference. Mechanisms of transmission. So, in other words, how do you get COVID? So, um, again, this is has changed a little bit, but for the most part, I think we have been pretty sure that these are respiratory droplets, possibly even aerosol related. So anyway, viral particles inhaled, touching mucous membranes, uh, with the virus on it. So anyway, multiple modes of transmission, but primarily you're thinking upper respiratory. And then it gets into quarantine. What is a quarantine? How long does it take? What's the purpose of quarantine? Um, why are they in a 14-day quarantine? Remember this, the first 6 months or so, it was 14 days was the kind of the quarantine baseline. That's been loosened a little bit to even 7 to 10 days, depending. So, but anyhow, you can speak to that as well. Even how things have changed or grown or progressed, or maybe not grown, maybe regressed in certain areas. Okay. Do you think these two gals are at risk for having COVID or testing positive for COVID? Again, these some of these questions are what you would say are fairly subjective. So this is another one that you could kind of put your own opinion out there. There's no right or wrong answer. Uh, Maybe they will, maybe they won't. Fact is, um, they're young, pretty healthy. I know early on they were pretty confident that the younger population had less chance or were less likely to contract it. We know now that that's not necessarily the case. That young people, if you're immunocompromised, no matter how old you are, you're potentially at risk, especially if you're not wearing the covering or practicing social distancing or other hygiene guidelines that are recommended. So anyhow, part two gets into kind of the structural aspects of COVID-19. So this is pretty fascinating. Talks a little bit about the RNA, the difference between RNA and DNA. So so anyhow, getting more into the nitty gritty now. What are the structural and functional differences between DNA and RNA? And of course, we know DNA is made up of the nucleotide or nitrogenous bases A, C, G, and T, right? So we find it in the nucleus. Naturally exists as a kind of a double helix. Um, and whereas RNA, and that those double helix strings of peptides are going to be connected or linked by hydrogen bonds. Now, with RNA, we know that those are single strands, and they are generally kind of secondary structures to DNA. They have what we call a ribose as its sugar, as opposed to a deoxyribose sugar. So that has to do with having an OH group attached to it. And instead of A, C, G, and T for the bases, we have A, C, G, and U. So the T and the U are going to connect when we start doing our translation. So let's see. Ba-ba-ba-ba. I don't know if there was anything else I really wanted to say about it. Oh, I think maybe DNA is the genetic material coding for the proteins as well as for RNA. We generally too would find RNA kind of all over the place, but definitely in the cytosol or the cytoplasm of a cell. So many functions for RNA. It's a kind of a cellular messenger. It has to do with catalyzing or speeding up chemical reactions inside of a cell. Anyway, found throughout the cell. And again, this COVID is the RNA group. So you'll be asked kind of a few questions about that. It says list one. Let's see. I don't know if one is considered a list, but name one or list multiples human viruses that have DNA-based genomes. Smallpox, herpes, right? You get herpes, you've got it forever. There's some papoviruses and adenoviruses. So anyway, you look up DNA-based human viruses. Next one asks about a glycoprotein. What is a glycoprotein? Well, we've talked about it a little bit at the beginning. Glyco, of course, means sugar. Protein having to do certainly with that structural peptide or string of amino acids. That's typically going to be, so a glycoprotein is going to be a protein with some glycos or some sugars coming off of it, some sort of carbohydrate chain that's going to be attached to that polypeptide or protein sequence. So protein is the kind of the root of this term, glyco being kind of the piggybacker or the attacher of the main protein. So glyco meaning carbohydrate, protein meaning a protein. So anyway, little carbs attached to long chain proteins. And oftentimes we'll find these on the surface of a cell. So we look at that phospholipid bilayer and we may have a glycoprotein coming off of that. And oftentimes it's going to be some sort of marker for cell to cell communication. We can see some attachment principles or actions occurring with glycoproteins connecting with other glycoproteins. They can help with protection, recognition, uh, just general cellular interactions. Okay, because again, you think about what's on the cellular membrane. There's a lot of these. We have the phospholipid bilayer, but we also have these little membrane channels. And then on top of that, we have a lot of glycoproteins kind of coming off of that phospholipid bilayer that again aid with communication as well as many other functions. All right. Why coronavirus may have glycoproteins on their membranes? Well, I think that the recognition and binding aspects to the human cells, these spikes that we see fusing that virus envelope to our cell membranes, probably would show that we've got some sort of, and there you can see the envelope and then these little spikes coming off. And then there's these little membrane glycoproteins kind of mixed in. And those are gonna those glycoproteins along with the spikes are gonna help with kind of that that binding and fusing of the virus to our host cells. Let's see. And what's the purpose? Again, to survive. They're trying to connect. They're trying. The virus is on the verge of survival, and it needs to attach to and connect with your cells. So these glycoproteins are going to aid in the connecting with the host cells. All right. So let's see. What else do we have? Phospholipid. Oh, we looked at phospholipid and phospholipid bilayer. Again, a phospholipid is a phosphate, which is going to be hydrophilic with a hydrophobic lipid tail. And we see so the bilayer is going to be those two lipid tails kind of coming toward the center of that cell membrane. So the outer and inner portions of the cell are going to be the phosphate and the phosphate. And then coming toward that bilayer is going to be where we find those hydrophobic lipid tails. Now, let's see, two rows. Again, the polar heads face outward, non-polar heads, the lipids are non-polar, going to be facing, that's hydrophobic. So no water in that tiny little membrane, that bilayer of that phospholipid cell membrane. And soaps generally have a polar head group and a non-polar tail, just like our phospholipids. Soap molecules tend to solubilize the hydrophobic proteins and phospholipids in the membrane of the COVID. So the COVID virus. So it pulls them apart and incorporates them into what we call the myceles. So this MICELLES. When we look at lipids and we look at emulsification, we see these little tails kind of going around these individual globules or droplets of fats. So anyway, it's possible that soap can break that wall or that barrier, that phospholipid barrier, and convert it into little tiny mycel structures that are going to, not allow that the nucleoid protein of that COVID to survive. So again, that's why washing your hands with soap is a big deal. We know that about all viruses too, right? So that's nothing new regarding soap and killing viruses. So again, it goes through a couple other wanting to find out more information. She'd heard of HIV and she'd heard of other viruses, so she wanted to find out a little bit more about COVID. So she asked about the basic steps of a lytic infection typical to a DNA virus like herpes. So not an RNA virus um, like COVID. So your typical kind of what we call lytic virus lysing. The virus gets recognized by the host cell at the cell membrane level, and then viral DNA gets released and it moves into the cell, and then it makes its way into the nucleus, and then that viral DNA gets kind of incorporated into the host DNA. And then we end up getting messenger RNA transcribed from the virus and sent out and translated by the ribosomes of the host cell. And then we start assembling the virus inside of the cell. And then the host cell eventually bursts and releases this kind of assembled virus. So these DNA viruses have to get through the nuclear envelope. They have to get through into the nucleus. So oftentimes our immune system is going to be able to to fend that off before it ever gets that far. If there's a foreign invader in the cytoplasm, hopefully our cells are going to recognize it and destroy it. This isn't always the case. So if it gets all, the virus gets all the way to the nuclear envelope or the nucleus membrane, then it can certainly become one with the cell, if you will. Let's see. Then we look at SARS or COVID and how that's different. Again, viral RNA never leaves the cytoplasm. So we get this simultaneous replication going on over and over and over from the messenger RNA for translation. Uses its own RNA polymerases for RNA replication, but uses the host machinery for the translation of it. So the host ribosomes, the host rough ER is being used. So the factory basically is taken over and held hostage, and the virus is basically saying, hey, we brought our parts in, you're going to assemble them. And so that in essence is what happens. From there, it can use host transcription and translation machinery to, again, just continue to assemble new viruses. So basically these RNA viruses come in, they get past the cell membrane, they get into the cytoplasm of the cell. They don't have to get to the nucleus, and they've brought their RNA with them, and then they take up shop in the with the ribosomes and the rough ER, and then anyway, things get out of control, or can. Increased infection rate. Well, that increased infection rate, faster evolutionary, we can evolve a lot faster in that area in the cytoplasm with the RNA. Higher mutation rates of the RNA because you don't have that DNA proofreading going on. So there's a little more checks and balances with the DNA. So RNA, maybe higher mutation rates. So we would see a lot more mutations or variations of an RNA virus versus a DNA virus, which is what we've seen, right? We've seen several different mutations or strains or variations of COVID. So part of that, again, is because it is RNA specific. Central dogma in biology is simply that DNA is replicated to make more DNA using its own DNA polymerase. And the polymerase breaks off. It's an ACE, right? It's an enzyme. So DNA gets transcribed into messenger RNA by RNA polymerase in a process called transcription. So we transcribe or write out the recipe that we need, and then that RNA gets translated into some sort of protein strand or enzyme or something by the ribosomes and the rough ER. So coronavirus, again, defies the central dogma because it doesn't need that DNA, doesn't need to get into the nucleus, doesn't need to affect the DNA. It makes its own RNA into more RNA, translates using our own ribosomes and rough ER. So there's no need for any type of DNA template. Let's see, you don't need to do that table. Hey, and then the unfortunate diagnosis. So you can read through that. It's got a list of medications that you're going to kind of research just a little bit. That you don't have to go too deep into these. These are very common antivirals used to treat flu viruses, HIVs, as well as herpes and a few others. So anyway, let's see. Oh, what's an antibiotic and why would that maybe be ineffective towards treating COVID? Again, antibiotics go after bacterial infections. We're dealing with a viral infection. So antiviral treatments, there to treat, they're, they're usually most effective early on, right? So maybe it slows the rate of viral progression. So antivirals in the early stages generally are going to be a lot more effective. And again, you can read through for number 3 for each of the antivirals why they won't necessarily work for COVID. And again, these could be somewhat subjective. Just kind of use your own background that you have. Again, I don't want you to have to feel like you need to go too far. And then again, using what you've learned about COVID, possible targets or target steps, consider the life cycle of COVID. So I think figuring out how to prevent the attachment, right? Like that, if we can prevent any virus from attaching to our host cells, well, there's the answer right there. So how do we do that? Well, good luck, right? Coat all of our cells in a special coating with something. But anyway, it gets into the linking of the corona spikes with our own ACE, angiotensin converting enzyme to receptor sites. If we can protect that, the first, so we can work our way even further out, right? We need to protect the first line of defense. So protecting the mucous membranes, the nasal passages, sinus membranes, or upper respiratory epithelium, washing hands properly, another first line of defense. So again, ounce of prevention worth a pound of cure. And that's what we saw with folks who were practicing social distancing. We're washing their hands. We're wearing masks. We're taking these precautions, not really engaging in a lot of travel and a lot of these hot spot areas, so like airports, churches, restaurants, bars, cafes, um, those types of places tended to be cruise ships, of course. These tend to be the hot spot, and still, from the very beginning and to now, that's pretty much been kind of the idea is, if you can prevent it from even entering the body by you know anyway, you guys know all this. You've heard it all, but humans are funny, man. You can tell them to do things, and they'll maybe do it for a little while. But if it doesn't fit into their daily routine, or if it doesn't fit into their schema, and it's a barrier, or it's a hurdle. Sometimes they just want to push right through it and keep doing what they're going to do, and nobody's going to tell them any different. So, so, I mean, COVID or no COVID, it's good to social distance, good to wash hands, and hell, wear a mask if you're in certain situations. But so anyway, hopefully you enjoy that case study. It gives you a little bit of. Obviously, it applies to right now. It's very modern to what we're dealing with. So anyway. Let's see. And you don't have to do number 5 or part 5. Okay, you don't need to do part 5. All right. So PhysioEx, again, I'm not going to spend a heck of a lot of time doing it. The beginning part, I'm going to do the activity with you, so you have that information if you're unable to perform the activity for whatever reason. And again, when we go back into Blackboard and we look at the PhysioEx hyperlink, we scroll down and we find serological testing, competency 6. Click on that. It's going to open up what you're supposed to do. And again, this hyperlink will take you right to the PhysioEx website. I do have the Word doc in there with the questions on it. So again, you're more than welcome to use this document. If you are having issues performing physio. So anyway, that'll just open a Word doc. I'm not going to switch screen sharing settings, but you'll be able to see that will download the Word doc. I do prefer the PDF from doing the actual lab simulation. But again, I know many of you have computer issues, and So anyway, you have access to those review questions that I grade, and that's really all you need to submit. So I'm just going to run through the, pardon me, I'm just going to run through the actual lab itself. I need to share a new screen for some reason. So, well, let me show you what's happening here. Maybe it'll work now, but I can go to PhysioEx and I can click activity 3 and maybe it'll work. No, and it just spins and then stops spinning and nothing's there. So, whereas if I go up here to the one above it, that one spins and that one pops up. So, if you're having issues with PhysioEx, please let me know. Another one thing I was able to do was open it in. I'm in Google Chrome, so I went into Internet Explorer, typed in PhysioEx, and it opened up fine. So I'm going to switch over to Internet Explorer. You know, Firefox may work as well for you, but try a different browser if you're having issues. So screen sharing. Hopefully that's the right screen. Let me make sure. Yes, it looks like it is. All right. So again, this is PhysioEx 12, exercise 12. This is activity 3, indirect enzyme linked immunosorbent assay. So that's a mouthful. That's why we call it the ELISA. Um, the first section goes through and talks about why we use it. It can test for the presence of an antigen. Remember, antigens are surface markers on cells, and antibodies are going to be in the serum of of the fluid or of the body. So typically we think of the antibody being in the plasma if we're talking about blood, and the antigen is going to be cellular. So a direct ELISA can test for antigens, the specific antigens, whereas an indirect ELISA, which is what we're going to do, is going to test for detection of antibodies. So if the patient has developed antibodies against antigens from whatever the pathogen is or pathogens are, that indirect ELISA can tell us whether the person has developed antibodies. So indirect, we're going after the antibodies. Direct, we're going after the specific foreign substance. We're going after the virus. We're looking for the antigens on the surface of the virus or bacteria. Okay, whereas indirect, we're looking for the antibodies that you produced to fight off the virus. So positive ELISA is going to mean that seroconversion has taken place. And again, that just means that the person has gone from testing negative for the antibodies to now testing positive. So they didn't have the antibodies in their serum. They've converted to now having antibodies in their serum. That's seroconversion. So they would test positive for the indirect ELISA test. So anyway, we use big micro titer plates. Many of you are going to take, if not all of you, are going to take microbiology. So you'll be working with 96 well micro titer plates. So they'll be coded in the direct test. So we're going to do the indirect ELISA test. So anyway, you can read through that. That's more or less kind of FYI. In the indirect, the 96, which again, what we're doing, we're testing for antibodies. The 96 well microtiter plate is going to be coated with antigens. The patient's serum sample gets added to the test for the presence of antibodies that bind to the antigens on the plate. So a secondary antibody will get added with an enzyme linked to it that binds to the constant region of the primary antibody if it is in fact present in the patient sample. So the constant region of an antibody has the same sequence of amino acids within a class of antibodies. So all IgG antibodies, Ig means immunoglobulin. This is immunoglobulin G. Antibodies would have the same constant region. The variable region of an antibody provides the diversity of antibodies and is the site to which the antigen binds. Okay, so Okay, so the variable region of the antibody provides the diversity of antibodies and is the site to which the antigen binds. So the configuration that forms in the indirect ELISA is antigen, primary antibody, secondary antibody. Okay, just as a direct ELISA, the addition of substrate is used to determine whether the sample is positive for the presence of the antibody. Okay, so we're going to use indirect ELISA to test for the presence of antibodies made against HIV. So this is a common HIV test. You'll use positive and negative controls to verify the results. Now, uh, we could have indeterminate. There's a range of indeterminate. When you do have an indeterminate result, it could mean the patient was recently infected and they haven't created enough antibodies yet to be considered to be positive. So they would get retested. All right, so let's skip through those. All right, let's get to the test. Now it's pretty straightforward. We're going to drag our 96. This one runs pretty well. So there's no clicking of necks. It's just going to move it along for the most part for us. So we're going to put our micro titer plate here, and then it says to drag the multi-channel pipetter to the pipette tip dispenser. Okay, and then we're going to draw up the HIV antigens. Oops, let me just, there we go. It's just a little. Let me see if I can. There. No, it doesn't really let me shrink things too terribly much. So I'll kind of be going up and down so you can see it. So anyway, so we put the HIV antigens in the titer plate. The simulation compresses to 14 hours. So we need to set the timer now to 14 hours and let that sit. Oops. Of course, we don't have to wait 14 hours, right? So it takes about 10 seconds. Okay, the incubation time allows the antigens to stick to the plastic wells of the microtiter plate. So we had to coat the plate with the HIV antigen. So we had a brand new microtiter plate. We're coating that plate with HIV antigens. All right. So drag the washing buffer squeeze bottle to remove excess antigens that didn't get stuck to the plate. So we're going to rinse off the plate a little bit, take it over to the sink. Then we're going to dab it a little bit, dry it off, and then send it back over there. Okay, so drag the 100 microliter pipetter to the tip dispenser to place a tip onto the pipetter. So here's our pipetter. So we're going to go over here, get a tip on it, and then we're going to go to this is a positive control sample. So this person this is a control that's HIV positive, a control that's HIV negative. And then we have patients A, B, and C. So we're going to get that positive HIV. We're going to put that in the first hole, dump that, and then it's going to do the rest on its own. So then the second well spot, it's going to put the negative controls, and then we're going to do patient A, patient B, and patient C. Okay, so again, we had an empty, brand new well plate. Coated the plate with HIV antigens, and then kind of rinsed it off, dried it off, and then in each of the circles, we're going to add. I don't even know what the question is. I'm just clicking that so we can keep moving. All right, set the timer to 1 hour, then click start. Okay, so then we added the patients or the positive HIV positive, HIV negative controls, and then patients A, B, and C to the plate. So now we need to rinse off a little bit. So they sat for an hour, and then we rinse off. Oops, and then we have to dry off our well plate, put it back, okay, and then we're going to do our multi. Okay, then we're going to go to the developing buffer. Okay, so then we're going to take that developing buffer We're going to add that in. And what that's going to do is activate and get key substance. So that's going to have that enzyme link to it. I shouldn't have answered that for you. But anyway, so we're going to set the timer to an hour. Click that so that developing buffer kind of gets that secondary connector or antibody going. And I believe, yeah, we got to rinse it off again. And take it over to the sink. Pat that baby dry. Bring it back over, and then it's going to ask us to do basically what we've been doing. And we're going to drag it over to the substrate bottle. That's what, if we've got a positive test, that's what everything's going to stick to. We're going to get our multicolors, and here's our wells. So we're just going off of this first column. So this would be the control. This would be control for positive, control for negative, patient A, patient B, and patient C. So what we're going to do is select each well, and then the optical density number is going to show up over here. So I'm going to click that. So right now we're just going to record data. We're not worried about any numbers. We click that, and we'll talk about what the numbers are. So let's just collect our data. Here's patient A. And again, it's looking at that optical density, patient B, and then patient C. All right, so let's look at the numbers before up here, the ranges. So if we get that an optical density of less than 0.3, then the person is HIV negative. Okay. If we're at a 0.3 to 0.499, then that's an indeterminate test. Again, it could mean they are recently exposed and they've started developing antibodies. And then greater than 0.4, so 0.5 or higher is going to be a positive test. So now just by looking at the so we don't, if we didn't even do optical density, our machine broke, but we're able to view with our eyes. Notice the ability for light to pass through. That's what this optical density means. So notice our control for positive, and then our patient C down here. They look very similar to one another. Now notice our negative control and our patient A. Those also look very similar to one another. And then we've got our B, which isn't quite the same as A or the negative control. So it's not quite as dark or as light colored, as high of a density as our positive and our patient C. So anyway, I'm not trying to spoil any of the fun here for you, but let's take a peek. So again, if we're above 0.5, that's a positive. So our positive control, good, we did it correctly, we mixed our chemicals properly, added things at the right time. So if the reason we do a positive and a negative, we do both positive control and negative control is because we want to make sure that our serums are correct, our HIV antigen, developing buffer, substrate, everything we're doing is going to make sense with and along the way too with each of the patients. If I do a positive control, I should see a very high positive because I already know it's the control group for a positive. So if maybe my developing buffer wasn't at the right temperature, or maybe it somehow got contaminated, or the substrate got contaminated, and we see our positive controls at 0.2 or something. Come up here, 0.2, that's a negative, not a positive. So we would have to redo the whole thing if that were the case in a regular lab. Now, so anyway, above 0.5 is a positive, definitely. Now, below 0.3 is negative. So their negative control ended up being 0.15. That's what we would expect. That's a good thing. Now we look at patient A, and we see patient A is 0.054. So they virtually have nothing in the way of antibodies against HIV, which is what we would expect to see if someone was HIV negative. So we see again anything under 0.3, that's way under. So that definitely is a negative. Now, if it's between zero point 0.3 and zero point 0.5, that's indeterminate. So patient B, which again, we kind of see their color doesn't really match the positive or the negative control. So patient B sitting at 0.43, they're going to be indeterminate. So they're going to indeterminate. So they're going to have to come back. Now, patient C, looking a lot like the positive control group, 1.99. That's well over 0.5. They would be considered a positive. And that's it. Congratulations. You did your lab. So you submit, submit, submit. And again, you can go through and do these questions if you'd like. You're not required to. These are the ones you're required to do. Again, this one has 4 questions. It just asks the difference between direct and indirect ELISA. Pretty straightforward. Why might a test be indeterminate? Again, we've discussed that as well. If the negative, what if our negative control was a 0.42, so it showed up as indeterminate? With some we bungled somewhere. We've got some sort of contamination going on, maybe with the buffer, maybe with the substrate. Hard to say, but either way, yeah, the whole point of doing the positive negative is to make sure all of our equipment and all of our chemicals are what they should be, right? That they're functional. All right. So anyway, let's see. And then lastly, the basic structure of antibodies. What are antibodies and why do we need them? Okay, so that's it for PhysioEx. Let's go back real quick over to our course. Okay, so case study 6 is done. PhysioEx is done. Now, module 2, video lectures and PowerPoints. That's where you're going to find the PowerPoints lecture for today, and then a 2-part video. So it's a little beefier, a little bulkier. Again, those are the review questions for the exam. You may want to have those out and ready while you're looking through, viewing, or watching the videos. And again, with the PowerPoints, I do have notes down here, so you can certainly read through those as well. Those will help you with the review questions as well. So anyway, that's all I have for today. I'll put the links to these videos as well in the YouTube this video. But again, you guys at this point, I think, know where to pretty much find everything. And again, next I'll have another video for module 3, unit 7, which starts next Wednesday. I'll have the videos up for that. Now let me look real quick at our course calendar. We do spend a couple of weeks on unit 7. So again, that does give you some time. It is a little beefier. There's one case study for unit 7, but we do have 2 PhysioExes. So I'll go through those. This says PhysioEx 6. It's really PhysioEx, I guess, maybe 5 point. I'll just put 5. So you're going to do 2 from PhysioEx 5. So anyway, next week will be a video that'll cover the next 2 weeks, basically. So anyway, I'll get all the videos out for you for each week and or each unit, and you guys can continue forth with those. So anyway, again, as always, if you have questions, please email me and I'll get back to you as quickly as I can. And again, those of you in Elkhorn, I hope to see you guys all in class next week, March 10th. And again, if you can't make it, shoot me an email and let me know. Other than that, have a great week, everybody.